Interferon. Interferons, IFNs, /Interferon/ are a group of signaling proteins made and released by host cells in response to the presence of several viruses. In a typical scenario, a virus-infected cell will release interferons causing nearby cells to heighten their antiviral defenses. IFNs belong to the large class of proteins known as cytokines, molecules used for communication between cells to trigger the protective defenses of the immune system that help eradicate pathogens. Interferons are named for their ability to interfere with viral replication by protecting cells from virus infections. IFNs also have various other functions. They activate immune cells, such as natural killer cells and macrophages. They increase host defenses by upregulating antigen presentation by virtue of increasing the expression of major histocompatibility complex (MHC) antigens. Certain symptoms of infections, such as fever, muscle pain and flu-like symptoms, are also caused by the production of IFNs and other cytokines. More than 20 distinct IFN genes and proteins have been identified in animals, including humans. They are typically divided among three classes: type I IFN, type II IFN, and type III IFN. IFNs belonging to all three classes are important for fighting viral infections and for the regulation of the immune system. Types of interferon. Based on the type of receptor through which they signal, human interferons have been classified into three major types. In general, type 1 and 2 interferons are responsible for regulating and activating the immune response. Expression of type 1 and 3 IFNs can be induced in virtually all cell types upon recognition of viral components, especially nucleic acids, by cytoplasmic and endosomal receptors, whereas type 2 interferon is induced by cytokines such as IL-12, and its expression is restricted to immune cells such as T cells and NK cells. Function. All interferons share several common effects. They are antiviral agents and they modulate functions of the immune system. Administration of type I IFN has been shown experimentally to inhibit tumor growth in animals, but the beneficial action in human tumors has not been widely documented. A virus-infected cell releases viral particles that can infect nearby cells. However, the infected cell can protect neighboring cells against a potential infection of the virus by releasing interferons. In response to interferon, cells produce large amounts of an enzyme known as protein kinase R, PKR. This enzyme phosphorylates a protein known as IF2 in response to new viral infections. The phosphorylated IF2 forms an inactive complex with another protein, called IF2B, to reduce protein synthesis within the cell. Another cellular enzyme, RNOSA-L, also induced by interferon action, destroys RNA within the cells to further reduce protein synthesis of both viral and host genes. Inhibited protein synthesis impairs both virus replication and infected host cells. In addition, interferons induce production of hundreds of other proteins, known collectively as interferon-stimulated genes, ISGs, that have roles in combating viruses and other actions produced by interferon. They also limit viral spread by increasing p53 activity, which kills virus-infected cells by promoting apoptosis. The effect of IFN on p53 is also linked to its protective role against certain cancers. Another function of interferons is to upregulate major histocompatibility complex molecules, MHCI and MHC2, and increase immunoproteasome activity. All interferons significantly enhance the presentation of MHCI-dependent antigens. Interferon gamma, IFN gamma, also significantly stimulates the MHC2-dependent presentation of antigens. Higher MHCI expression increases presentation of viral and abnormal peptides from cancer cells to cytotoxic T cells, while the immunoproteasome processes these peptides for loading onto the MHCI molecule, thereby increasing the recognition and killing of infected or malignant cells. Higher MHC2 expression increases presentation of these peptides to helper T cells. These cells release cytokines, such as more interferons and interleukins, among others, that signal to and coordinate the activity of other immune cells. Interferons can also suppress angiogenesis by downregulation of angiogenic stimuli deriving from tumor cells. They also suppress the proliferation of endothelial cells. Such suppression causes a decrease in tumor angiogenesis, a decrease in its vascularization and subsequent growth inhibition. Interferons, such as interferon gamma, directly activate other immune cells, such as macrophages and natural killer cells. Induction of interferons. Production of interferons occurs mainly in response to microbes, such as viruses and bacteria, and their products. Binding of molecules uniquely found in microbes, viral glycoproteins, viral RNA, bacterial endotoxin, lipopolysaccharide, bacterial flagella, CpG motifs, by pattern recognition receptors, such as membrane-bound toll-like receptors or the cytoplasmic receptors RIG-I or MDA5, can trigger release of IFNs. Toll-like receptor 3, TLR3, is important for inducing interferons in response to the presence of double-stranded RNA viruses. The ligand for this receptor is double-stranded RNA, dsRNA. After binding dsRNA, this receptor activates the transcription factors IRF3 and NF-kappa B, which are important for initiating synthesis of many inflammatory proteins. RNA interference technology tools such as dsRNA or vector-based reagents can either silence or stimulate interferon pathways. Release of IFN from cells, specifically IFN gamma in lymphoid cells, is also induced by mitogens. Other cytokines, such as interleukin-1, interleukin-2, interleukin-12, tumor necrosis factor and colony-stimulating factor, can also enhance interferon production. Downstream signaling. By interacting with their specific receptors, IFNs activate signal transducer and activator of transcription, STAT, complexes. STATs are a family of transcription factors that regulate the expression of certain immune system genes. Some STATs are activated by both type I and type II IFNs. However, each IFN type can also activate unique STATs. STAT activation initiates the most well-defined cell signaling pathway for all IFNs, the classical Janus kinase STAT, JAK-STAT, signaling pathway. In this pathway, JAKs associate with IFN receptors and, following receptor engagement with IFN, phosphorylate both STAT1 and STAT2. As a result, an IFN-stimulated gene factor 3, ISGF3, complex forms. This contains STAT1, STAT2 and a third transcription factor called IRF9, and moves into the cell nucleus. Inside the nucleus, the ISGF3 complex binds to specific nucleotide sequences called IFN-stimulated response elements, ISRs, in the promoters of certain genes, known as IFN-stimulated genes ISGs. Binding of ISGF3 and other transcriptional complexes activated by IFN signaling to these specific regulatory elements induces transcription of those genes. A collection of known ISGs is available on Interferon, a curated online database of ISGs, www.interferon.org. Additionally, STAT homotimers or heterodimers form from different combinations of STAT1, -3, -4, -5, or -6 during IFN signaling. These dimers initiate gene transcription by binding to IFN-activated site, GAS, elements and gene promoters. Type I IFNs can induce expression of genes with either ISR or GAS elements, but gene induction by type II IFN can occur only in the presence of a GAS element. In addition to the JAK-STAT pathway, IFNs can activate several other signaling cascades. For instance, both type I and type II IFNs activate a member of the CREAK family of adapter proteins called CRKL, a nuclear adapter for STAT5 that also regulates signaling through the SIG/RAP1 pathway. Type I IFNs further activate P38 mitogen-activated protein kinase, MAP kinase, to induce gene transcription. Antiviral and anti-proliferative effects specific to type I IFNs result from P38 MAP kinase signaling. The phosphatidylinositol-3 kinase, PIEK, signaling pathway is also regulated by both type I and type II IFNs. PIEK activates P70S6 kinase 1, an enzyme that increases protein synthesis and cell proliferation, phosphorylates of ribosomal protein S6, which is involved in protein synthesis, and phosphorylates a translational repressor protein called eukaryotic translation initiation factor 4E binding protein 1, IF4EB1, in order to deactivate it. Interferons can disrupt signaling by other stimuli. For example, interferon alpha induces RIG-G, which disrupts the CSN5 containing 9 Colombian pesos signalosome, CSN, a highly conserved multiprotein complex implicated in protein denatylation, deubiquitination, and phosphorylation. RIG-G has shown the capacity to inhibit NF-kappa B and STAT3 signaling in lung cancer cells, which demonstrates the potential of type I IFNs. Virus resistance to interferons. Many viruses have evolved mechanisms to resist interferon activity. They circumvent the IFN response by blocking downstream signaling events that occur after the cytokine binds to its receptor, by preventing further IFN production, and by inhibiting the functions of proteins that are induced by IFN. Viruses that inhibit IFN signaling include Japanese encephalitis virus, JEV, dengue type 2 virus, DEN2, and viruses of the herpes virus family, such as human cytomegalovirus, HCMV, and Kaposi's sarcoma-associated herpes virus, KSHV or HHV8. Viral proteins proven to affect IFN signaling include EBV nuclear antigen 1, IBNA1, and EBV nuclear antigen 2, IBNA2, from Epstein-Barr virus, the large T antigen of polyomavirus, the E7 protein of human papillomavirus, HPV, and the B18R protein of vaccinia virus. Reducing IFN alpha activity may prevent signaling via STAT1, STAT2, or IRF9, as with JEV infection, or through the JAK-STAT pathway, as with DEN2 infection. Several pox viruses encode soluble IFN receptor homologs, like the B18R protein of the vaccinia virus, that bind to and prevent IFN interacting with its cellular receptor, impeding communication between the cytokine and its target cells. Some viruses can encode proteins that bind to double-stranded RNA, (tRNA), to prevent the activity of RNA-dependent protein kinases. This is the mechanism rehavirus adopts using its sigma-3 protein, and vaccinia virus employs using the gene product of its EEL gene, P25. The ability of interferon to induce protein production from interferon-stimulated genes, ISGs, can also be affected. Production of protein kinase R, for example, can be disrupted in cells infected with JEV. Some viruses escape the antiviral activities of interferons by gene, and thus protein, mutation. The H5N1 influenza virus, also known as bird flu, has resistance to interferon and other antiviral cytokines that is attributed to a single amino acid change in its non-structural protein 1, NS1, although the precise mechanism of how this confers immunity is unclear. Interferon therapy. Diseases. Interferon beta-1a and interferon beta-1b are used to treat and control multiple sclerosis and autoimmune disorder. This treatment is effective for reducing attacks in relapsing remitting multiple sclerosis and slowing disease progression and activity in secondary progressive multiple sclerosis. Interferon therapy is used, in combination with chemotherapy and radiation, as a treatment for some cancers. This treatment can be used in hematological malignancy, leukemia and lymphomas including hairy cell leukemia, chronic myeloid leukemia, nodular lymphoma, and cutaneous T-cell lymphoma. Patients with recurrent melanomas receive recombinant IFN alpha-2b. Both hepatitis B and hepatitis C are treated with IFN alpha, often in combination with other antiviral drugs. Some of those treated with interferon have a sustained virological response and can eliminate hepatitis virus. The most harmful strain, hepatitis C genotype I virus, can be treated with a 60 to 80% success rate with the current standard of care treatment of interferon alpha, ribavirin, and recently approved protease inhibitors such as Telaprevir, Incivek, May 2011, Boceprevir, Victrolis, May 2011, or the nucleotide analog polymerase inhibitor Sofosbuvir, Sovaldi, December 2013. Biopsies of patients given the treatment show reductions in liver damage and cirrhosis. Some evidence shows giving interferon immediately following infection can prevent chronic hepatitis C, although diagnosis early in infection is difficult since physical symptoms are sparse in early hepatitis C infection. Control of chronic hepatitis C by IFN is associated with reduced hepatocellular carcinoma. Unconfirmed results suggested that interferon eye drops may be an effective treatment for people who have herpes simplex virus epithelial keratitis, a type of eye infection. There is no clear evidence to suggest that removing the infected tissue, debridement, followed by interferon drops is an effective treatment approach for these types of eye infections. Unconfirmed results suggested that the combination of interferon and an antiviral agent may speed the healing process compared to antiviral therapy alone. When used in systemic therapy, IFNs are mostly administered by an intramuscular injection. The injection of IFNs in the muscle or under the skin is generally well tolerated. The most frequent adverse effects are flu-like symptoms, increased body temperature, feeling ill, fatigue, headache, muscle pain, convulsion, dizziness, hair thinning, and depression. Erythema, pain, and hardness at the site of injection are also frequently observed. IFN therapy causes immunosuppression, in particular through neutropenia, and can result in some infections manifesting in unusual ways. Drug formulations. Several different types of interferons are approved for use in humans. One was first approved for medical use in 1986. For example, in January 2001, the Food and Drug Administration, FDA, approved the use of PE-gylated interferon alpha in the USA. In this formulation, PE-gylated interferon alpha-2b, pegintron, polyethylene glycol is linked to the interferon molecule to make the interferon last longer in the body. Approval for PE-gylated interferon alpha-2a, Pegasus, followed in October 2002. These PE-gylated drugs are injected once weekly, rather than administering 2 or 3 times per week, as is necessary for conventional interferon alpha. When used with the antiviral drug ribavirin, PE-gylated interferon is effective in treatment of hepatitis C. At least 75% of people with hepatitis C genotypes 2 or 3 benefit from interferon treatment, although this is effective in less than 50% of people infected with genotype 1, the more common form of hepatitis C virus in both the US and Western Europe. Interferon-containing regimens may also include protease inhibitors such as boceprevir and telaprevir. History. Interferons were first described in 1957 by Alec Isaacs and Jean Lindenman at the National Institute for Medical Research in London. The discovery was a result of their studies of viral interference. Viral interference refers to the inhibition of virus growth caused by previous exposure of cells to an active or a heighten-activated virus. Isaacs and Lindenman were working with a system that involved the inhibition of the growth of live influenza virus in chicken embryo chorioallantoic membranes by heighten-activated influenza virus. Their experiments revealed that this interference was mediated by a protein released by cells in the heighten-activated influenza virus-treated membranes. They published their results in 1957, naming the antiviral factor they had discovered interferon. The findings of Isaacs and Lindenman have been widely confirmed and corroborated in the literature. Furthermore, others may have made observations on interferons before the 1957 publication of Isaacs and Lindenman. For example, during research to produce a more efficient vaccine for smallpox, Yasuichi Nagano and Yasuhiko Kojima, 2 Japanese virologists working at the Institute for Infectious Diseases at the University of Tokyo, noticed inhibition of viral growth in an area of rabbit skin or testis previously inoculated with UV-inactivated virus. They hypothesized that some viral inhibitory factor was present in the tissues infected with virus and attempted to isolate and characterize this factor from tissue homogenates. Independently, Montoho, in John Inders's lab, observed in 1957 that attenuated poliovirus conferred a species-specific antiviral effect in human amniotic cell cultures. They described these observations in a 1959 publication, naming the responsible factor viral inhibitory factor, VIF. It took another 15 to 20 years, using somatic cell genetics, to show that the interferon action gene and interferon gene reside in different human chromosomes. The purification of human beta interferon did not occur until 1977. YH10 and his co-workers purified and produced biologically active, radiolabeled human beta interferon by superinducing the interferon gene in fibroblast cells, and they showed its active site contains tyrosine residues. Tan's laboratory isolated sufficient amounts of human beta interferon to perform the first amino acid, sugar composition and N-terminal analysis. They showed that human beta interferon was an unusually hydrophobic glycoprotein. This explained the large loss of interferon activity when preparations were transferred from test tube to test tube or from vessel to vessel during purification. The analyses showed the reality of interferon activity by chemical verification. The purification of human alpha interferon was not reported until 1978. A series of publications from the laboratories of Sidney Pestka and Alan Waldman between 1978 and 1981 described the purification of the type I interferons IFN alpha and IFN beta. By the early 1980s, genes for these interferons had been cloned, adding further definitive proof that interferons were responsible for interfering with viral replication. Gene cloning also confirmed that IFN alpha was encoded by a family of many related genes. The type 2 IFN gamma gene was also isolated around this time. Interferon was scarce and expensive until 1980, when the interferon gene was inserted into bacteria using recombinant DNA technology, allowing mass cultivation and purification from bacterial cultures or derived from yeasts. Interferon can also be produced by recombinant mammalian cells. Before the early 1970s, large-scale production of human interferon had been pioneered by Kerry Cantel. He produced large amounts of human alpha interferon from large quantities of human white blood cells collected by the Finnish blood bank. Large amounts of human beta interferon were made by superinducing the beta interferon gene in human fibroblast cells. Cantel's and Tan's methods of making large amounts of natural interferon were critical for chemical characterization, clinical trials, and the preparation of small amounts of interferon messenger RNA to clone the human alpha and beta interferon genes. The superinduced human beta interferon messenger RNA was prepared by Tan's lab for Cetus Corporation to clone the human beta interferon gene in bacteria, and the recombinant interferon was developed as beta-seron and approved for the treatment of MS. Superinduction of the human beta interferon gene was also used by Israeli scientists to manufacture human beta interferon. Human interferons. Teleost fish interferons. Teleost fish interferons. Teleost fish interferons.